Thank you for the introduction. So my name is Trista Mckenzie. I'm from the Department of Geology and Geophysics at the University of Hawaii at Manoa, and I'm going to be talking today about the influence of surface and groundwater inputs on water quality in Kaneohe Bay. So let's get started. First, I want to talk about what I will be discussing during this talk, so I'm going to do a brief overview. First, I want to talk about our previous knowledge, the background on the mechanism, and the role of groundwater fluxes in Kaneohe Bay, specifically looking at recent research in the last couple of years and seasonality related to that. Then I will be talking about current research, our new results on groundwater discharge in the Kahului region, and how we want to take this research and go further with it into our future research plans, looking at the central and south sectors of Kaneohe Bay. So starting with an introduction and motivation, one of the previous research has shown that there is a notable seasonal variation between groundwater dominance and surface water runoff dominance into the bay. So during our dry season, we are seeing that groundwater makes up more of the percentage of the total volume of water versus during our wet season, when surface runoff is the dominant component of the total volume of water into Kaneohe Bay. So we have a graph here, and on the x-axis we have months and years, and on the y-axis we have millimeters per month volume water. And you can see with precipitation in the dashed gray line, surface runoff in the blue line, and then groundwater flow with the black line, that during the wet season, precipitation is much higher, and this is outlined in the red box. And so then surface runoff is much higher. And on the flip side, during the dry season, the summer months, we see the opposite. The precipitation goes much, decreases, and we see a change in the proportion of that means that groundwater is the more dominant component. So what is groundwater discharge? And I'm going to use the term groundwater discharge and submarine groundwater discharge somewhat interchangeably. So groundwater discharge, this is infiltrated water that travels through porous media, and then due to the hydraulic gradient or downward slope pressure, it gets pushed out along the shoreline between the sediments. And this is a really important and, I guess, less previously quantified element to what's making up our water components within the bay. And so it's really important to quantify it. There's been research that's shown on Pacific Islands that it can comprise 3 to 4 times the amount of surface runoff. And this has been previously undocumented or compartmentalized. So it's really important that we look at this because it takes on the characteristics of whatever land use is in the surrounding area. This diagram also illustrates the other process we are looking at, which is surface runoff, which is dominantly driven by precipitation. So looking at our study area more specifically, historically, Kaneohe Bay has been just divided into 3 main sectors: the northwest sector, the central sector, and the southern sector. The northwest sector is subject to 52% runoff into Kaneohe Bay. The central sector is 39%, and the southern sector is 8%. And so we will specifically, in our study, be looking at 3 selected watersheds within the 3 greater main sectors. And so specifically in the northwest sector, we're looking at the Kahului watershed. In the central sector, we're looking at the Shayle watershed. And in the southern sector, we're looking at the Kaneohe watershed. So I'm going to delve into something a little more specific now, and this is going to be more related to our most recent research efforts. The Kahului region and the lagoon. It is fed by 3 streams, the Ahui Manu, the Kahului, and the Waihe streams. And We are teasing out the differences in the components of groundwater versus surface runoff contributions into each stream, and then also what is in the lagoon. The lagoon is a really important area to study because it's been subject to ongoing beach closures by the Hawaii Department of Health because there have been ongoing high bacterial levels. And we suspect that these ongoing high bacterial levels are due to the fact that there is a poor understanding between surface and groundwater fractions. So it's important to then tease out the differences and the components fed by each. The objectives of this study. One, we want to map surface and groundwater inputs into the Kaneohe watershed and Kaneohe Bay. And we are doing this using chemical elements characteristic of groundwater, specifically radon and radium. And these elements are enriched in groundwater due to surface area differences between that and surface water. So we are able to tell the differences between groundwater and surface water. Within these components, we want to estimate the natural and anthropogenic inputs. So specifically, we're looking at nutrients and pesticides at the moment. And then we are doing this project in collaboration with two other projects. One you'll hear more about later today, talking about thermal infrared imaging with Michael. And then last week, Chrissy gave a talk about her research on the analysis of water for septic and cesspool biological tracers. And so we hope with these three projects that are working together that we can kind of generate a holistic picture for what's really going on in this region. So on to some initial results. We've been working hard to map the Kahaluu area. And this is a map of the groundwater discharge spatial distribution indicated by radon. So we have found definitive elevated groundwater inputs, especially in the Ahuimanu stream, which there's a very small arrow indicating it, and then also in the Kahaluu streams, and in the lagoon itself. So this tells us this is an important element to look more closely at. But not only is the groundwater elevated within the stream channels, it's also elevated along the coastline, which I will show further in a second. And these groundwater inputs show an alternative and not previously documented pathway for nutrients and pollutants to impact coastal waters, particularly in this area where there are agricultural and on-site disposal systems that could influence the characteristics of the groundwater. I guess another reason to think about why this may be important is that Kaneohe Bay has a barrier reef, and elevated groundwater fluxes in other areas have shown that this can lead to ocean acidification and negatively impact these reefs. So it's a very important quantity to characterize. I wanted to focus a little bit more on the coastline, just so I've recolored the same map as previously to show really the variation along the coastline. The relative groundwater concentrations along coastlines are different than within the stream. And so, yeah, you can definitively see that there are hotspots for groundwater inputs along Kaneohe Bay near Kahaluu. And these groundwater inputs are similar in magnitude to a study conducted along the coastline 4 years ago. So this is an ongoing problem, and we need to further look at it. So what are our next steps? Because this research is new. I started in the fall, so this is an ongoing project. We want to, one, show seasonality in ground versus surface water dominance and the associated nutrient and pesticide fluxes with these. So we want to take measurements during the dry season and the wet season, and also have numbers for the nutrient and pesticide fractions within these, within the ground and surface water components. And then also we want to expand to the central and southern sectors of Kaneohe Bay so that we can provide bay-wide estimates of groundwater's role on nutrient fluxes. So in conclusion, previous research has demonstrated a strong seasonal variation between ground and surface water dominance in Kaneohe area. And Groundwater is an important and potentially major component in coastal waters in Kaneohe Bay, and we have confirmed the presence so far and show the spatial heterogeneity of groundwater discharge in the watershed. Our next steps will build on previous findings and continue to look at seasonality and mechanisms of submarine groundwater discharge in Kaneohe Bay. I just wanted to quickly thank my advisor, Dr. Henrietta Dulai, my fellow students who have helped me in the field, the Hawaii Department of Health for their consultation and help with this project as well, and then the University of Hawaii Sea Grant for funding this project. Thank you.
Cool. Thanks for coming today, everybody. My name is Shayle Matsuda. I'm a graduate student at the Hawaii Institute of Marine Biology. And my talk today is called Stuck in the Middle with You, What the Future Holds for Corals and Their Symbiotic Algae. So the first time I went scuba diving, 8 years ago, and I descended into the water, and I really distinctly remember how I felt. As soon as your face hits the water, it's this rush of really cold feeling. And I remember looking down and just seeing a whole new world, different than anything I could have imagined before. I was seeing like walls of corals, I was seeing tons of fish just moving around, and something that really grabbed me was this big feeling of discovery, but also the curiosity. How do these systems work together? What were all of these organisms, and what were the things that I wasn't seeing? So the marine environment has a lot of similarities with land, right? Here on Oahu, we have different neighborhoods. We have different neighborhoods that are that have their different sorts of resources. So you have like in your one neighborhood, you have like maybe your school systems, your public health institutions, and many of us have our homes. Some of us stay close to home, but a lot of us travel around to different environments to get energy, to go work out, to go to our jobs, to go shopping for food. But what we can see is that there's a lot of similarities and differences with neighborhoods that are pretty close together. I'm just going to highlight a few of those right now. So we have our inhabitants, right? On the far left, you can see that we in our neighborhoods have our locals. We also have a lot of visitors who come to our neighborhoods too. Same thing is true in the marine environment and coral reefs. We have a lot of different organisms who stay put and others who you see moving around more. We have our health centers here where we go when we're not feeling well. Coral reefs also have those kind of things too. On the right, you can see a cleaning station where we have some cleaner rafts that are picking parasites off of some fish. We have different types of schools. This is a little farther of a stretch, but on the left we have our public institutions where we go. Everyone's following the same direction, and on the right you see a schooling of fish who stick together to avoid predation. And then we have our structures, and our structures can tell us a lot about these different environments, right? Just on the far left, you can see two pretty distinct structures. One, we've got these tall buildings and downtown, and then we've got these more suburban areas right over here. And then in coral reefs, we have structures that are really important too. And these structures are they rocks? No, they're corals. When I tell people that I study corals, a lot of the time I hear, oh, those are those things that I cut myself on when I'm surfing, but there's a lot more to them. These structures play a really large role in our coral reef communities. But people who think that they're rocks are actually not so far off, because right under the surface layer, the coral tissue is a hard skeleton, which you're seeing right here. So right here, right under these thin tissue layers is this calcium carbonate skeleton, and this skeleton is what actually builds the structures that form our coral reefs. And I'm going to talk about these a little bit more today when I get into my research. So the coral reef you can think of like a neighborhood. It has our coral structures, it has its residents, it has influxes of energy and outputs like that. And these neighborhoods can be close together and experience some of the same challenges, but they can also be very, very different. So my research is in Kaneohe Bay, right up here. And just like the island of Oahu with its neighborhoods, Kaneohe Bay has over 50 patch reefs marked in these little yellow numbers right here. And these patch reefs or neighborhoods are all very distinct. They have their residents, they have their structures. There's a lot of movement between these reefs, but what happens on the reef is also very unique for the organisms that live there. And that's where I do my research on corals. But before I dive in a little deeper, I want to kind of get us all on the same page about some coral biology. So what are corals? Corals are animals. And this is a zoomed in image right here of coral, so ice coral. And what you're seeing here are these individual polyps, and each of these polyps is an individual animal. So when you hear people refer to the coral colony, what it is really a collection of animals that all live together. And we can kind of think about this structure is like an apartment complex. So each of the coral polyps that I showed in this last picture right here, these guys, each of these coral polyps we can think of as an apartment, an identical apartment in this coral colony or this complex. And everything is the same. So it's not like you live in your one apartment and then like your relative is the next apartment. It'd be like a whole apartment of just you. So what happens when corals are born, you have a single coral polyp that then buds off to create its neighbor. And that process continues and continues and continues as the coral then builds itself. So these apartments are really important. And inside of these apartments, that's where things start to get interesting for me. So I'm really interested in how these corals associate with their symbionts or their partners, these single-celled algae right here. You might hear them called zooxanthellae or symbiodinium, but what they really are these single cells that live inside each of these coral polyps or little apartments right here. And just like land plants, what happens is these algae are able to take advantage of the energy coming down from the sun, photosynthesize just like land plants, and then they take that energy and actually convert it onto their coral host. But it is a two-way street, so while the algae is doing this and feeding the coral is also providing food that the algae needs as well as protective environment, protective housing. Now this isn't just any sort of like best friend kind of partnership. This is an obligate partnership, meaning that without these symbionts, without these algae, coral polyps cannot survive. And you might have heard something about this before, climate change and stress. So we have humans, we have a big impact on the environment. This has to do with the way that we create energy, our transportation system, the way we create food. And a lot of these methods can have drastic effects on the ocean environment. So increased sea surface temperatures, changing ocean chemistry, or even things like increased sedimentation, which is like literal dirt washing into the ocean, can have a really negative impact on coral reefs and these coral environments. And specifically, all three of these different stress factors are really bad for this partnership between the symbiotic algae and the corals themselves, and causes a breakdown in that relationship. So you guys, I'm sure no one here's a strange coral bleaching. So what happens is when there is a stressor in the environment and this symbiotic relationship is broken down, the coral can no longer maintain that relationship with its algae, and the algae are expelled or leave the coral colony. And the algae are really important because they also give the coral its color. So this is a healthy looking coral through the microscope on your left right here. You can see all these polyps. And on the right, you can see a bleached coral, or a coral that has expelled its symbiotic algae. And it looks white, which oftentimes folks will see white coral and they're like, oh no, that one looks like it's dead, but it's not dead yet. Instead, you can see all of these individual polyps right here. So the coral is still alive, the polyps are still here. They are able to feed on plankton that's moving around in the water system and survive for a certain amount of time. And if that stressor is removed, they're able to reform that partnership again with those algae. However, if the stressor continues, then they will often die. And that happens with a lot of these major coral die-offs that a lot of you have been hearing about in the news. So here's an image from the Huffington Post, and what you can see on your far left is a healthy coral reef, nice brown color because of those algae. Surface warming temperature, so we have coral bleaching here in the middle. The corals look white. And when the corals are white, that means the tissue is still there, so all hope is not lost. However, if the conditions don't return to normal soon enough and the corals are under too much stress and they do die, what you'll see is that this reef environment will then become overgrown and the macroalgae, the bigger algae, will kind of start taking over. And so it's when you see this kind of more fuzzy, dead-looking reef, that's when you know that we've hit the point of no return. So something that I'm really interested in is the neighbors. You can be in the same neighborhood, you can be in the same coral reef ecosystem, you can be the same species or type of coral. There can be the same stress event, and then you have one neighbor totally fine, the other neighbor completely bleached. Why does that happen, right? So what we're learning is that different algae, like different types of algae, are able to have different stress response to these stressors. And they're able to give that resilience over to their coral host. So for example, the way that we classify these different types of algae, it's not in terms of species types, but more that, we look at them in terms of clades. So like clades A, B, C, D, closely related, but definitely very different. And what we've seen recently in our lab is that corals that associate with what are known as like clade C type algae are less tolerant to stress, meaning those are the ones that are more likely to bleach. Whereas corals that associate with more clades D are more tolerant and less likely. And so what I'm doing for my one of my preliminary research studies is I'm looking at how variable are these different types of algae within one single coral colony. Because what we know is that certain types of coral can have associations with one type, another type, or sometimes even multiple types at the same time. So what kind of variability are we finding in some of these individual colonies, these different complexes? How does the environment around them impact that? So does water flow, does light coming down, does even the shape of these corals influence what types of algae are living there, and that therefore that kind of stress resilience to those stressors? And then finally, what would this impact be on the corals bleaching themselves or not, and then also on the recovery? When they do get those symbionts back, are they the same types? Do more opportunistic symbionts move in? What does that tell us about those types of things? And so my study is focusing on a really common reef-building coral in Kaneohe Bay called, it's a rice coral called Matsuda capitata. And what's really interesting about this coral is that it is really, really dynamic. You have these really nice plate morphologies here to these very branching structures. Sometimes you have a little bit of both, as in this one down here. There's also two color morphs. We see more of like a dark brown morph over here, and then this kind of more rusty orange color morph. And also what's really interesting about these ones is that these corals associate with algae in clade C, clade D, and sometimes both. And so why are they doing that? What is the difference between which types are where, and are they there in the same types of density? So this is the species that I'm working with. And the way that I'm going to be doing this landscape study is I'm going to be using this new technique called structure from motion photogrammetry, which is basically a fancy way for saying I'm creating really cool 3D models of corals to look at these questions. And so the way you do that is I take a couple hundred photographs all around the coral colony, and then using software, I put these images together to create my 3D model. So all of these blue rectangles up here represent one of the camera angles that I took those pictures from. And so you can kind of see, like right here, I've got a lot of coverage. Over here, probably could have used a little bit more. But what I can do here, let's see if I can get here to the internet, is when I stitched all of these together, I'm able to create a 3D model that looks like this. And let's see if I can zoom. There we go. Zoom in. And what this allows me to do is actually see and measure the structural geometry and take a look at, what would the differences be in an environment somewhere like right in here where it's really densely branched versus like these tips of this colony right here that get a lot of light. And what this model is going to let me do is actually be able to measure some of these angles, some of the flow rates and irradiance and things like that. Let's see, hopping back over here. Do I have to go through this whole thing again? There we go. All right. So, well, what I'm really interested in is this 3D mesh, as cool as the models are. And so what this mesh is going to allow me to do is basically track random points on the colony. I'm going to go in there and sample 50 to 100 points for genetic sampling, which will let me essentially sequence the DNA and figure out what the identities are of those algae in these different environments. So like the parts that are not getting a lot of light, the tips of the coral that do get a lot of light, the areas that are kind of underneath these branches. And some of our research partners in the Caribbean have done work similar to this. And what they're finding is that even in an area that is from like just from the tip of something down to the side a little bit, they're actually seeing a lot of differences in the identities of these algae. But we haven't done anything like this on any of the species that are out here. And so this is going to be a really interesting study for us to see if our corals are doing the same sort of thing and what kind of variation we're finding out. So I'm just in the preliminary phases right now, but what you're looking at right here is this is a single plate from one of the colonies in the field. This is the part of the colony that is furthest from the center, and this is from where the plate was broken off. This is the top of the plate, and this is the bottom. And you can see all these little white sections are where I took tissue samples from. And you can see that white structural skeleton underneath. And what you're looking at is I ran these samples twice for each tissue sample, and everything that is in gray came back as clade D, and everything in blue came back as clade C. So what we can tell right away from here is that this plate is a very clade D dominated plate. But you can see from the areas that were closer to the actual body of the colony, that's where we're starting to see more instances of this blue, of this clade C. Now, what can that tell us? Does that have to do with maybe those areas were more shaded, maybe there was less flow? Those are sort of the questions that I'm looking at answering. And maybe this will tell us a little bit more about, how do these corals prepare for bleaching? Like, is having one dominant type a better strategy? Is having multiple types a better strategy? Those are sorts of the types of questions that I'm hoping to answer. But pulling back out from the single colony scale back to our neighborhood and our bigger environment, picture of understanding coral biology, understanding how the climate is changing and impacting our coral reefs, and why these neighborhoods and the marine environment are so important. And I'd like to leave you with these sort of three main points. Coral reefs and coral colonies individually are really important for biodiversity. They're the home for a lot of our juvenile animals. A lot of our larger organisms in the ocean come to the coral reefs to have their offspring. Really good providing a shelter for those juveniles to grow up as well. The coral reefs are also in that sense, seeding for a lot of the fisheries and the food sources that we have and a lot of our own energy. And then finally, coral reefs are really important for coastal protection for here on Hawaii and a lot of our other coastal environments, providing a big break for a lot of the energy coming in from the main ocean. So they are very important not only for the biodiversity and ecology in the oceans, but also for us here on land as well. And with that, I would like to say thank you to all of my partners and to all of you for coming today. And at the end, I'll be happy to take any questions. Thanks.
All right, so my name is Michael Mathioudakis, and I'm a graduate student at UH Manila in the Geology and Geophysics department. My advisor is Craig Glenn, and I'm working in the Coastal Geology Research Group, studying discharges into Kaneohe Bay. So Trista and I are part of the same research groups. I know she touched on a little bit of this. But Kaneohe Bay is the largest sheltered body of water in the Hawaiian Islands, and it contains one of two barrier reefs in the Hawaiian Islands, with the other barrier reef on Molokai. And basically a barrier reef will surround a bay or lagoon type enclosed body of water, absorb a lot of wave energy, and allow a lot of biodiversity. And it has naturally oligotrophic waters, which means there's very low nutrient levels. So the Kaneohe Bay area naturally has low nutrient levels, but it has had increasingly exceeding nutrient levels due to human sources of nutrients, which I'll get to later. And since the bay is on the windward side of the island, it gets a lot of rainfall. So the main problem here in the bay is harmful bacteria in the water. There's Enterococcus and Clostridium, as well as nutrient inputs from cesspools and agriculture. as Trista talked about earlier, some of these beach zones are either closed off or have warning signs posted because of the high levels of bacteria here. So the two main human impacts on Kaneohe Bay are thought to be OSDS systems, which stands for on-site disposal systems, and agriculture. Both of these are high sources of nutrients into the bay. Cesspools and septic tanks are the two primary forms of on-site disposal systems. And there are a lot of on-site disposal systems on Oahu. It's actually the only state that allows cesspool construction. For those of you who are not aware of what a cesspool is, it's basically a big pit on your property that will store all the wastewater from your home. There are estimated to be 14,000 on-site disposal systems just on Oahu, with a very high density of these systems, with some areas containing 350 for a single square mile. The highest density of these systems are along the coast. As you can see from this image, the areas in red are areas where there's a high density of these systems. And Kahalu is a very OSDS dense area. It's within the Kaneohe watershed and is a main focus area of our study. So even though nutrients are naturally occurring and essential to life on Earth, excess nutrients, especially in a bay like Kaneohe that has naturally low nutrient levels, can cause invasive algal blooms and coral bleaching. And it's a compounded problem in Kaneohe because of the high density of on-site disposal systems, and because of the high level of rainfall on the side of Oahu, and because of the sensitive bay ecosystem. As you can see from this map here, the rainfall level in the Kaneohe watershed area is much higher than on the leeward side of the island. And because of that rain, there's a lot of flushing of these on-site disposal systems, and leaking of these systems will then cause this contamination to move into the bay. So most of you have probably seen some sort of diagram like this before describing the water cycle. So when we get a lot of precipitation, it will infiltrate into the ground and move to the water table, where there's standing groundwater, saturated groundwater. And while this water infiltrates, it can pick up contaminants from septic systems and from cesspools. And once that water gets into the groundwater, it can move on to the ocean via either submarine groundwater discharge, what will flow directly from the groundwater into the ocean, or via flow into streams, where the groundwater will flow into the streams and then eventually reach the ocean. So either way, nutrients from these septic systems or cesspools can find their way into the ocean once they leak out from within these systems. So submarine groundwater discharge is a mix of fresh rainwater and recirculated seawater. Basically, this water from the coast will move in towards the land and will mix with the groundwater that's coming out from the water table. And so it creates this somewhat salty water that's less dense than seawater. So the submarine groundwater discharge will float on top of the seawater. And there's actually more water discharged by submarine groundwater discharge than by rivers. So you don't see submarine groundwater discharge, but it's very prevalent in the coastal hydrologic cycle. So here's another diagram of submarine groundwater discharge. The purpose of our study is basically to determine how connected these on-site disposal systems are and these sources of nutrient pollution to Kaneohe Bay. So how does this contamination move from the land to the bay, and what are the effects of that? What is the transport like? We're trying to identify these zones where this is most occurring, basically find the source areas and figure out how it's getting into the bay, so that legislators can use this information to regulate cesspool and septic tank construction and use. The main method that we're using to find submarine groundwater discharge is via thermal infrared sensing. We're using drones, and basically groundwater is colder than surface water. Surface water is sitting in the sun all day, so it has this solar heating, whereas groundwater is insulated by the earth. So when it discharges out into the coast, it's naturally colder than the ocean water, and you can detect these small temperature differences with infrared sensing. Did a couple pictures here of us out in the field doing these drone flights. This is the drone that we use. We have our thermal camera right there, and we fly at an altitude of about 120 meters. That allows us to get beneath the clouds and get a good clear image. We actually get a pretty wide swath width, so one single image covers 100 meters of the coastline. Usually on one of these flights, we're able to get about 1,000 meters in distance. Here's a stitching of our study area. This is the Pallu Lagoon. There's not a whole lot of groundwater discharge going on in this image, but you can see that along the edges of the land, the color is a little darker blue, so there's colder water there. There's little groundwater seeps coming in. Again, this groundwater discharge is variable seasonally, as Trista talked about, and can vary with the tides as well. As the water level of the lagoon in the bay drops, more groundwater will flow into the bay. In the top left corner there, you can see kind of a picture of some submarine groundwater discharge. This is at a low tide, and the groundwater is seeping out, and you can actually see it in this location. So a lot of times if you put your feet into the sand when you're at the beach, you can feel the cold water, and that is actually submarine groundwater discharge. So this is an image from a test flight. As you can see, the darker spots, the purple spots, are where the groundwater is seeping out. And in addition to the infrared sensing, we're also doing sampling of groundwater wells, of streams, and of ocean water. And that allows us to further track the nutrient and bacteria loading. So we're testing for nutrients, specifically nitrogen, phosphorus, and silica, and for bacteria and Clostridium. And then also carbon and nitrogen stable isotopes. Carbon and nitrogen will decay based on their residence time and based on their sources. And water isotopes as well will decay. So when you have some precipitation, it will degrade the longer it's in the water cycle, and you can use those water isotopes to track how long water is, basically how young rainwater is. So we're also using groundwater modeling, and we calibrate that with our field data. So we will go to groundwater wells and collect water levels. We will collect temperature, pH, water quality parameters like that, and use that to calibrate our model. And also doing stream gauging, so we can gauge the sections of streams that are being influenced by groundwater, so sections of streams that are gaining water from groundwater and sections of streams that are losing water from groundwater. And that's all I have. Thanks to my advisor, Dr. Craig Glenn, and to Dr. Henrietta Dulai, Trista's advisor, who's also vital in our research group, and the University of Hawaii Sea Grant for their funding. And I'd like to thank the Department of Health. They're very involved in this project. Robert Whittier, Watson Okubu, and Scott Murkawa. I'll take questions with everyone.
Here. Okay. Questions for our speakers? Any questions on any of the talks that are heard? Yes. Go ahead.
Groundwater depth. You're measuring radon. You're also looking at bacterial levels. Radon levels are basically the rate of discharge, contemplating the volume of discharge. Because if you have a quick rate of discharge, the rate
So we are doing snapshot studies. We are also looking at flow, and we are taking end members using piezometers. So we are trying to capture a very holistic picture, but we're also looking for the continuous picture.
But the time frame wise, you're looking at, if you have a sudden storm, huge amount of discharge.
Of course. And we are taking weather influences, particularly precipitation and storm runoff, into considerations. Right now, our measurements have not been taken anywhere near those kind of events. We are putting I guess a continuous monitor that's in the form of a buoy called the sniffer that measures radon, and we will have time series measurements, and that should capture any storm fluxes or other weather when maybe we don't want to go out and risk our lives during the higher flow events.
And conversely, when you look at material, same thing. Immediately after a storm, we might have high bacterial levels.
That would be anticipated, yes.
And then it will drop. So I don't know what kind of time values you would have over so many days or weeks.
We will be modeling in the later stages of the research. Right now, this is very preliminary.
I think he's asking about your sampling frequency.
Yeah, that's what it's-
Oh,
How often are you getting a lot of samples?
right now we are there on a weekly or bi-weekly basis. So it's
Any other questions?
Question about the core. So what's the difference between clade C and clade? What's the difference between clade C and clade D? And is one, have you noticed one is more prone to white conditions than the other?
Yeah.
And is that a function of temperature or other, you said temperature, but I wonder if there are other environmental factors.
Yes, and it's all complicated. So the first question, so there's we call them clades. You can kind of think of them like species, but we haven't, like taxonomically, we haven't, the work hasn't been done to make those delimitations about like what's a species or not yet. So they're kind of like groups or types.
So is it based on DNA? Or?
Yeah. Yeah, so we have phylogenies, and within those clades that are A, B, C, D, there are subclades. You can have like D1 or C15, things like that, that are more closely related. And there is work being done to try to figure that out right now. But things that are like subclades are more closely related. So C and D are pretty different, which is, I know, not answering your question exactly, but they're different, and potentially could be species different later on when we're there. But what we have found is that in different species of corals, certain symbiont types, certain types of these clades are showing, like those particular individual corals are not bleaching as much as their other ones, and we're not sure to what extent that role has to do with the algae themselves versus like the genotype of the host, the coral host itself. So we're looking at a lot of those different kind of factors too. But like when the bleaching event happens, we have folks in our group have tagged corals all over Kaneohe Bay and have been taking tissue samples through the bleaching events that just happened over the last couple of years. And from those, we're finding that corals that are more dominated by C were more likely to have bleached. And so like using that prior knowledge, we're then kind of, I'm more interested in looking at the individual level, because like a lot of these individual corals they'll be dominated by a certain type, but we don't know to what extent they're dominated by that, because a lot of times coral biopsies will just be one little piece taken off of a tip, and we're not necessarily how representative of the entire individual organism that is.
What about the variability of its own value?
So that's what I'm curious about, exactly. Some coral species have a really tight-knit relationship with their zooxanthellae. Those tend to be vertical transmitters, meaning that the parent will pass those algae on to their offspring. Horizontal transmitters, meaning they're born without that association, they get it from the environment. And so we're kind of looking at those relationships too, are the ones that are more tightly knit, that are not able to necessarily, exchange out symbionts that are maybe more resilient or not able to be as responsive. Are those more likely to be resilient versus the ones that potentially have that option? My hypothesis is that if a coral is able to associate or has associations with multiple clades, that in some of those clades are more stress resilient, that might give that coral a leg up, and that's kind of what it is that I'm looking at.
So clade is strictly the coral, right?
Clade, when I talk about clades like C and D, that's the algal symbionts. And so that's a symbionts. And so like with some of the stuff that the other studies we're looking at now, we're repeating these kind of studies with different coral genotypes or coral individuals that way. And so we're taking that into consideration, and then on top of that, taking those environmental factors that you're mentioning as well. So we're doing temperature stress studies, we're doing ocean acidification studies, and we're measuring things like flow and sedimentation, because it's hard because as far as coral biology goes, something that's so interesting about it is that it's really complex. It's really hard to just say we're only looking at this one thing. So you have a lot of times you've got to take into consideration the genotype of the coral, the individual coral itself, the algal symbionts. There's also bacterial symbionts in there that we also are looking into what role those play as well. But then also taking all the environmental factors. So a lot of the studies we've done have been on stress related to temperature, but now what we're moving into is looking at the changing pH and how that's also affecting these relationships too. So that's kind of what a lot of this work is going to be moving more into. But it's kind of it's such a dynamic system that it's hard to give you like a very concrete kind of straightforward answer on that. So I apologize for that, but that's where we are.
I have kind of a follow-up question to that. You are at this point though seeing a distinct correlation between the clades and the degree of bleaching, is that correct?
In the particular corals that we're tracking in Kaneohe Bay, we are starting to see that coming through. A paper by Cunning et al. From our lab recently was the one who first kind of got the data to start seeing that. It's not 100% one way or the other, but we're starting to notice that correlation, so we're trying to investigate that a little bit further. It's hard because the corals, like in Kaneohe, for example, that are associating with these different types of algae, if these species exist in other water basins around the world, they might have different symbiont types. And so that's something else that I'm looking at. I'm looking at a separate project, how flexible are these corals? Like, can we expose them to different types of algae, and will they associate with those ones that might be more stress resilient or not? So we're kind of trying to see what their natural flexibility will allow them to do, because those are kind of the questions exactly that we're trying to really get at. Like, could this be a tool that we use in the future to help these corals be able to survive these increasing stressors? It's a great question.
One more quick question. Yeah. What about clades A, B,
et
cetera, so forth?
Mm-hmm. And so they are, yes, right? In Kaneohe Bay, which is where like the corals, there's only a few dominant coral species in general. And so the rice coral that I showed you, those ones associate with C and D primarily. There's another really dominant finger coral varieties out there that associates with clade C15, right? Another one of those subclades, and that one is very, very resilient. And so something that I'm actually looking at right now is, can we take those algae and expose them to the other species, and would they associate with them or not? And so we're not finding in those species that I study, they don't have associations with like as frequent with like A, I, G, H, those ones, but in other marine environments in the world, like those are very prevalent. And so it's hard, because what you want to be able to do, right, is like understand how those systems are functioning in each of these different places in the world. But then what I'm trying to do is see if there's anything that we can learn about how the flexibilities work in these systems here that then may or may not tell us about how these other systems might work. Yeah.
I'm sorry, can I have one?
Well, just a follow-up to that, I don't know if it's the same question or not, but how do you share your information with your counterpart researchers in other parts of the world? Like you take these hundreds of photographs of your coral, you have the, your DNA.
Yeah.
There's a ton of data that, your counterparts in Australia did, and now you're doing, and you want to match it up with what everyone else is doing.
Absolutely. Great question. Yeah. That's really, really important. And I think that I would say in general, a lot of folks at the Hawai'i Institute of Marine Biology at UH were really moving towards open source data. It's critical, especially given how fast climate change is becoming an impacting factor on a lot of coral reefs, that you can't necessarily sit on data for 2 or 3 years while you're waiting for publication, right? So you do have these publications are very important, but what we are seeing is a lot of inter-ocean basin collaborations. A lot of folks, like our lab included, are moving towards putting a lot of this data online. It's really accessible as soon as we get it. So on video from this lab, we have a link to our website where we're going to start posting a lot of this data coming up. So that will be available. All of the code that we use for processing this is going to be available via GitHub. But I think it's something that you're seeing across a lot of research labs worldwide, is like getting, especially things like sequence data, right, out there as soon as we can. Because you're right, like we do a lot of talking to other labs. So like, if we're like, okay, you're sequencing that genome, we're going to work on this one instead, and then we'll collaborate this way. Our lab, sister lab, is in Australia, the Hanauma lab, so we're working really closely with them to repeat a lot of similar types of studies and questions in both of these environments. So it's something that I think we're seeing a huge movement towards in general in coral biology.
Does that apply to you guys as well with your infrared images and all that? Is there a standard format for storing it and sharing it?
It's actually a really small community right now that's doing the thermal infrared. We're actually one of the first research groups to be doing it. There was a research group in Korea that was doing it, and they really are the only other ones right now. So we have had collaboration with them. And they were actually the ones who taught us how to use this technology, and so we're kind of the main research group that's doing it right now, hoping that it will expand out. And really, drone technology is expanding so quickly. There's new legislation every day. There's new technology every day, and so it's definitely something that we're on the cusp of right now, but that is definitely going to expand in the future. But yes, we're collaborating with those who are doing it. Yes.
There's not a standard yet, or
No.
We will be publishing our results on a website, I guess, to be determined, which is, I guess, not the best piece of information for you guys, but we'll let you know, I guess, when that materializes.
I have one question. At this point, since the issue is with the cesspools and septic tanks, and even if regulation is introduced to try to keep them from being manufactured and created, is there any thought given to coming up with a way to treat the harmful nutrients and bacteria at the source before it enters the groundwater system?
Yeah, I mean, that would ideally be installing septic tanks at these locations, because cesspools don't provide any sort of treatment, whereas septic tanks at least provide some. It's not the treatment you're going to get from pumping it to a treatment station, but it's more treatment than a cesspool. So yes, they're looking at replacing cesspools with septic tanks, or just hooking people up to sewer lines, which is a whole other issue. But cesspool is basically the most primitive of all the treatment types, and so upgrading even to a septic tank makes a big difference.
Last question, you guys. How much would that cost?
I think it's about $40,000 or $50,000
To like convert all of them?
to change one cesspool to septic tank. It's pretty expensive. Right now they're working on some tax credits. I think it's a $10,000 tax credit, but obviously $10,000 doesn't pay for a $40,000